Good evening, everyone. My name is Alex Subik. I'm the Deputy Vice Chancellor, Research and Development at this fine institution. I'm thrilled to welcome you all to Swinburne's 8th annual Barbara Dicker Oration. But before we start, I would like to respectfully acknowledge the traditional owners of the land on which we gather and pay respect to all Aboriginal community elders, past, present, and emerging, who have resided in the area and have been an integral part of the history of the city and the region. A special welcome to the Dicker family, the Honourable Ted Baillieu, former Premier of Victoria, former Chancellor, Emeritus Professor Ian Wallace, AM, Chancellor Professor John Pollaers, OAM, Vice Chancellor Professor Linda Christensen, AO, members of the Swinburne University Council, members of the Swinburne Executive, and of course all the other distinguished guests. Also a personal thank you and welcome to our international guest and this year's distinguished speaker, Dr. Gustavo Turecki. Welcome. Given that more than 20% of the population are affected by mental health problems every year, Swinburne is deeply committed to supporting our researchers to explore and understand the inherent complexities of neurological and psychological conditions. In fact, our investment and commitment to this field has also resulted in psychology and cognitive sciences discipline being awarded a ranking of 5 through the Excellence in Research Australia assessment this year, which is among the world leading in this category. Through Swinburne's Centre for Mental Health and our state-of-the-art neuroimaging facilities, our work on improving the mental health of individuals has seen us take significant steps towards creating a better future for all. We're currently making many exciting upgrades to our neuroimaging facilities to ensure we can produce faster, more precise and nuanced ways of measuring the brain's complex function and structure. Integral part of this strategy is establishing a new data science team within the facility to help drive new groundbreaking discoveries using also big data and machine learning. In early 2020, we will be refurbishing our facility with a focus on patient and carer comfort as we are opening the facility to a broader stakeholder base through our research collaborations with clinical partners and with leading research institutes in the Melbourne Biomedical Precinct. Recently, Swinburne has also launched a new access and support scheme designed to facilitate and support early career researchers to develop novel, cutting-edge interdisciplinary neuroimaging methods and to augment funds such as those generously provided by the Barbara Dicker Brain Sciences Foundation. Our students, staff, and researchers are very fortunate to work so closely with the Barbara Dicker Brain Sciences Foundation to not only bring together teams of divergent thinkers, but to deliver world-class research outcomes and innovations leading to profound social impact. We certainly have a wonderful evening ahead of us. And to begin, it is my pleasure to invite Professor Gavin Lambert, Director of our Iverson Health Innovations Research Institute, to present the research showcase. Gavin, thank you.
Thanks very much, Alex. As Alex mentioned, this is the 8th oration, and for that period of 8 years, the Barbara Dicker Brain Sciences Foundation have been supporting research into mental health here at Swinburne University of Technology. Importantly, they've been focusing the attention and support on early career scientists, to give those early career scientists that leg up to try and get their career started. So I'm very, very pleased to actually be highlighting and introducing 3 of those scientists to present to you today. So our first speaker, Dr. Wei-Lin To, and Wei-Lin is actually leading research into hallucinations and cognition in people with Parkinson's disease. Wei-Lin.
Good evening, everyone. Thanks for having me. So thank you, Gavin, for introducing me. I'm an early career researcher at the Centre for Mental Health, and I'm here to talk to you today about my study investigating hallucinations and cognition in Parkinson's disease. But before I begin, I'd like to take this opportunity to thank the Barbara Dicker Brain Sciences Foundation for their generosity in supporting my research. So I'd like to start by providing a bit of a background on Parkinson's disease. It's a neurodegenerative disorder affecting the cells in the middle part of the brain. These cells are responsible for producing a chemical called dopamine, which is necessary for the smooth control of muscles and movement. Parkinson's usually affects patients aged above 60, but an earlier onset is possible as well. The key symptoms of Parkinson's may be categorized into motor and non-motor symptoms. Although it's known as a movement disorder, non-motor symptoms can be prominent and include hallucinations, dementia, depression, or sleep problems. Our study will focus on these non-motor symptoms. Treatments currently available are mostly aimed at managing the more debilitating symptoms and can range from dopaminergic medications to lifestyle and environmental modifications. Now, I'd like to move on to what we know about hallucinations and cognition in Parkinson's disease. Hallucinations can be defined as unusual sensory experiences that occur in the absence of corresponding external stimuli. So, for instance, hearing voices or seeing images that others do not. Majority of hallucinations in Parkinson's disease takes place in the visual domain, and it can affect almost half of all patients. A typical hallucination often involves seeing a person or an animal walking around. It can last for up to a few minutes and usually occurs when the patient is fully alert. Some people are not alarmed by these experiences, whereas others perceive them as unpleasant and anxiety-provoking. Hallucinations in other sensory modalities have also been reported, but much less is known about these experiences. The majority of people living with Parkinson's also experience mild impairment in their cognition. So this refers to their attention, memory, and reasoning skills. When such decline becomes severe and is coupled with other symptoms such as delusions, we call this dementia. Importantly, the presence of visual hallucinations is known to be associated with Parkinson's dementia, depression, and other poor functional outcomes. So our current research in the area comprises a two-part study. Study 1 is theoretical and consists of a systematic literature review for hallucinations across each of the sensory modalities and related cognition. Study 2 is empirical and involves recruiting and interviewing 50 patients with Parkinson's with one or more modes of hallucinations. We collect information on the characteristics of these hallucinations, conduct a brief cognitive assessment, as well as record ratings of depression and sleep. We are now midway through this process and are on track to complete data collection at the end of the year. As it's currently a work in progress, I don't have any data to share with you today, but please watch this space. Thank you.
Thank you very much. Our next speaker will be Selma Music, and Selma is actually focusing her work on exploring attentional bias in patients with chronic post-traumatic stress disorder.
Thank you. Good evening, my name is Selma, and today, as Gavin said, I will be talking about post-traumatic stress disorder and attentional bias. Attentional bias to threat is the tendency to pay attention to threat in the environment while ignoring basically everything else. These three components have been identified in attentional bias to threat and have been linked to the development and maintenance of PTSD, post-traumatic stress disorder, which is an illness that sometimes people experience after experiencing a traumatic event. Various theories have been proposed to explain attentional bias to threat in PTSD. I will present two. The first one is the hypervigilance avoidance pattern, which suggests that people with PTSD tend to focus their attention to threat, and then they get overwhelmed and will avoid it. That's the first and third component. The maintenance pattern suggests that people with PTSD would identify the threat and then struggle to disengage from it. Most research to date has found support for that first hypothesis, the hypervigilance avoidance hypothesis. And this was measured using computer-based tasks where participants were asked to press a keyboard. We have investigated attentional bias a little bit differently, and we've used eye tracking. Now, what we've done is we had a large pool of small pool of refugees that they have experienced very severe trauma, and we separated them into 3 groups. PTSD group, a group of participants that have experienced war trauma but don't have PTSD, and a trauma-exposed healthy control group. And what we've done is we've asked them to look at images that present different emotional content. And I'll present to you the data of that. So this is a real participant. So what our studies have found is that individuals with PTSD tend to look at the trauma image, or tend to fixate on the trauma image a lot more than the other stimuli, and they tend to dwell on it for longer. Particularly so when that trauma image is congruent with their trauma experiences. So this is support for the maintenance hypothesis, which suggests that people with PTSD struggle to disengage from threat, and is similar to OCD and depression, or the attentional pattern there. So why is this research important? With the increased number of refugees daily, attentional bias modification training may assist underserviced and remote groups where face-to-face counselling and mental health care might not be available. I would like to thank the Barbara Dicker Brain Sciences Foundation for their help with this project. Thank you.
Thanks, Selma. And our final presenter for the showcase is Dr. Matthew Hughes, and Matthew will be exploring, or will be presenting on his exploration of neurocognitive impairments in people with a hoarding disorder.
Hello, my name's Matthew. We've been for a number of years now conducting some work on assessing the brain function of people with hoarding disorder, and specifically how they're impaired. Okay, so you may have guessed from this picture here, this is the bedroom, the living room of an individual with hoarding disorder. This is a typical sort of living space for them. So you may guess from that, or from reality television if you're into it, that they have a strong urge to both acquire and save items, and also they have a lot of distress when they are asked to discard those items. But what is surprising is the number of people who actually have this disorder. It's extremely common. And this is a big problem, not just to themselves, but also to others, not just for hygiene and so on, but obviously fire risks are an extreme danger. Hoarding houses, house fires are around 10 times more dangerous and burn more strongly than a normal house fire. So it's a big issue, and it's very difficult for the firemen to actually put the fires out just to get in there. So a lot of work's been done on trying to ascertain what sort of cognitive problems they have, and a number of things have been identified, including probably obviously decision-making and organisation, but also they have problems with attention and their ability to control their thought and behaviour. We call it cognitive control. I'll just go back slightly. We've been running this study with a cognitive task that tries to capture those problems. It's called the AX version of the continuous performance task. And we're doing that, having these people perform this in the MRI scanner, and also healthy individuals to compare them to. So this task, there's a number of trial types. The most important one, or not the most important one, but the most common one, starts with an A, and then, sorry, you're actually missing a line down the bottom, but anyhow, it doesn't matter. The task is, to respond, respond to an X only after it is presented after an A. So the X comes up immediately after the presentation of the A, and then another trial type is an A is presented, but then a Y is presented, but they're required not to respond to those Y stimuli. So this is our critical trial. So the AX trials, that's around 70% of trials. These AY trials is around 10% of trials. And then another trial type begins with a B, and then a Y is presented, and they're required to just ignore those stimuli. Okay, so we do a lot of these trials, and we compare the AY trials to the BY trials, and we get and then we compare those, the activation in those contrasts between healthy individuals and hoarding patients. So don't worry about the different brain areas. This maps onto what's called the salience network. So our conclusion is that hoarding patients exhibit impaired function in the salience network, which may underpin the other aspects of cognitive impairment that they have, and this can be due to altered dopaminergic transmission. Thank you.
Thank you very much to all the presenters. Ladies and gentlemen, that just gives you a bit of an idea of the breadth of work that goes on here at Swinburne and the breadth of the work that is supported by the Barbara Dicker Brain Sciences Foundation. Now, importantly, to put this into context and how important it is for the early career scientists to get this particular type of support, last week our National Health and Medical Research Council announced the individual support packages. Those salaries are what we used to term fellowships, and the success rate for the first time for early career scientists dropped down below 10%. So it is a very, very competitive world out there. We need all the support that we can give to our students and early career scientists. Many of that is coming through the infrastructure and the support from Linda and Alex and all the other staff at Swinburne, but imperative to that is the support from the community, support from foundations like the Barbara Dicker Brain Sciences Foundation. So on that note, I'll hand over to Dr. Tony Dicker, the current chair of the Barbara Dicker Brain Sciences Foundation.
Thank you, Professor Lambert, and thank you to those three wonderful presentations. It's just great to hear what's happening at the cutting edge of research here at Swinburne. Chancellor, Vice-Chancellor, distinguished guests, I'd like to extend on Professor Subik's words of welcome to this evening's oration. The Barbara Dicker Oration, or as my sister Jen and my brother Craig and I might call it, Mum's Oration, is an occasion where we get to hear some wise words. Mum passed away about 9 years ago, but she's always one to pass on some wise words to us. They weren't always the sort of wise words that we wanted to hear as teenagers or young adults, but wise they were anyway. In fact, I suspect that the wise words I can hear her saying to me now are, "Speak slowly." So the family were very proud when Swinburne 8 years ago offered the Barbara Dicker Brain Sciences Foundation the option of having this oration in her name. A platform to hear wise words from all around the world. The Barbara Dicker Brain Sciences Foundation was the brainchild of my father Ian, who was keen to provide funding for research into areas of depression, sleep and dementia. Swinburne has been an amazing partner in seeing his wishes come true. They've been so giving, so helpful, but also so practical in managing this philanthropic activity. So anyone here in the audience this evening who might be considering donations towards research, look, we as a family are very happy to recommend Swinburne as a partner in those endeavours. Wise words are why we're here tonight, which is why it gives me great pleasure to introduce tonight's main speaker to you. With our previous speakers this evening, we've set the bar nice and high for you, but I'm sure you'll have no trouble jumping over it. Professor Gustavo Turecki is both a MD and a PhD, so he's a doctor. As a clinician, he's a professor and chair of the Department of Psychiatry at McGill University in Montreal in Canada. That's over on the west side over there. Oh, the east side, sorry, over there. His clinical focus is patients with refractory major depressive disorders. But as well as doing his clinical work, he has a scientific hat. He's a scientific director for the Douglas Institute and director of the McGill Group for suicidal studies. The McGill Group has the Douglas Bell Canada Brain Bank, managing over 3,000 brains, which is one of the largest brain banks in the world. So he's in charge of a lot of important neurons there. He also holds a Canadian Research Chair in Major Depressive Disorder and Suicide. There's over 450 publications, so I won't go through them all. And for those of you who want to learn about how zinc finger protein in your prefrontal cortex can make you resilient to stress, the publication comes out of Nature Neuroscience in the next couple of weeks, I believe. So his focus is on the molecular changes in the brain that occur in major depression and changes that occur with treatment responses. He's also looking at the neurobiological basis of the suicidal brain. It's my great honour to welcome to the stage Professor Gustavo Turecki.
Thank you very much. Good evening, everyone. It's really a pleasure to be here. It has actually been a pleasure to spend a few days here with all of you. I was very warmly welcomed by everyone I met, and I had the opportunity to see the really innovative work being done here. I think that here there is some sort of a culture maybe, but there's a lot of things going on that it's different from what I've seen everywhere else, and I think that you've managed to really think outside of the box, and I want to congratulate all of you, and I hope you continue continuing on this path. So let me tell you a little bit about my work, and I'm gonna discuss basically early life adversity as imposed by childhood trauma, and how is that it may impact the brain and behavior. So early life adversity, childhood maltreatment, or childhood abuse, these are different names that we use for the most severe form of adverse childhood experience. So adverse childhood experiences denote a large group of negative experiences that children have that can go from losing a parent to being raised in a poor family to the most extreme being abused. There are four main types of childhood maltreatment. So physical abuse, sexual abuse, emotional abuse, and neglect. They often occur in combination. And what is important is that most of the perpetrators of the abuse are primary caregivers. And it seems that the identity of the abuser has an important impact on the consequences of the abuse. So abuse is unfortunately frequent. About 25% of adults report having experienced some form of abuse as children. In terms of sexual abuse, it's much more common in females than males. And many children, in fact, are exposed to abuse primarily in the early years of their lives. So in fact, it blows the mind to think that about 2.5 over 100,000 children die as a consequence of the abuse that they suffer from their caregivers. On the other hand, it also has an important economic impact. The economic impact is given by the direct consequences of the abuse and direct costs on the healthcare system, on the social support and youth protection system, etc., but also because of the indirect cost that it has directly emotional to the person who suffered abuse, as well as by the long-term consequences, as we're going to discuss in the next slide. It has a direct relationship with levels of poverty, so that the lower the education and the higher the poverty, the more likely it is to be present. So what are the consequences of childhood abuse? So first, injury. Individuals who were exposed to adversity early on in life are more likely to suffer different types of injuries through their lives. And you would see most of the consequences of childhood abuse have something to do with a tendency to engage in risky behavior and difficulties to actually control the person's own behavior. Opportunities, so people who were exposed to early life adversity are less likely to complete their education and have opportunities through life and career. Risky behaviors, as I said, so as a result of the risky behavior, people are more likely to have problems such as being infected with infectious sexually transmitted illnesses and to have car accidents and being involved in all kinds of accidents. Chronic disease, because they take less care of themselves as well, so they're more likely to have problems like diabetes, different forms of cancer, and actually die. They're more likely to die early on from physical causes as well. So infectious diseases such as HIV, as I said. Maternal health, so individuals who were exposed to life adversity are more likely to get pregnant early and have complications and problems of teenage pregnancy. And finally, negative mental outcomes. So I got involved in studying early life adversity precisely because of its relationship with negative mental health outcomes, and specifically its association with suicide. So I have been interested in understanding suicide risk through my career, and one of the things that we know is that individuals who die by suicide are more likely to have had histories of early life adversity. So here you have, for instance, how we understand suicide today, and we think that individuals who die by suicide on one hand have a given predisposition, and there are a number of different factors that contribute to that predisposition, one of which is the exposure to early life adversity. Now, most of the individuals who die by suicide do not have a history of early life adversity, but a significant minority does, about 40%. So why is that individuals who were exposed to early life adversity have all these long-term consequences, including increased lifetime risk of suicide? Yes? And this is the question that I'd like to discuss with you tonight. So, what happens? Why is that a traumatic experience early on in life can modify, negatively so many things in the lives of these people? So I'm going to give you a hypothesis, yes, of how is that we conceptualize this and how is that we think that this may actually happen. So first, to gain insight into possible mechanisms, we have been studying cohorts that are representative of the general population. So this is one cohort that we have. So this is a cohort that is representative of the normal population from Quebec, where I live. Quebec, it's one province from Canada. And in this cohort, we have 3,000 children that were recruited to the cohort when they were young, at the age of 6. So these children were interviewed, not themselves, but their teachers and caregivers were interviewed on a yearly basis from the age 6 to 12, and then at different periods of their lives, they were interviewed directly, and then we collected a lot of different information. So when we looked at this cohort, and then we asked questions about their development of traits, of behavioral traits and emotional traits. So we look, for instance, that children in this cohort can follow under different trajectories over time. Yes? So for instance, here on your left, you have how they develop in terms of anxiety traits. Yes? So being more likely, more prone to be stressed. So we see that, for instance, children, either they are very anxious at the age of 6 and remain very anxious until the age of 12, and then would likely remain also anxious later on. Some children are anxious, but then become less anxious. Some were less and become more, and some were not anxious at all and remain not anxious. Yes? So you have different types of developmental trajectories of anxiety, and similarly here for disruptiveness, which is a measure of being more hyperactive, aggressive, and oppositional in behavior. And what you see is that also here you have 4 different types of trajectories. So this type of study allowed us to look at how is that these behaviors predict suicidal behavior later on in life. So one important thing is that what we observed in these cohort studies is that the relationship between having been abused as a child and the risk of suicidal behavior later on at the age of 21, measure here, is in part explained by how they developed in terms of the anxiety trajectories and disruptive trajectories in such a way that it seems that the exposure to adversity early on, of exposure to abuse, triggers somewhat the development of high anxiety and high disruptive behaviors. So far, so good? Good. So we set up this hypothesis that we think that the way that exposure to early life adversity leads to suicidal behavior, and we can make the same parallel to all the other negative mental health outcomes, it's in part explained by this differential development of emotional and behavioral trajectories. Yes? All right. So then the question is, why? What happens? So what happens as a result of the experience of adversity that leads to this differential development, that leads to these kids to become more anxious and more impulsive and aggressive? All right. So our brain, it's a plastic organ, which means that it has the capacity to adapt with experience. And it changes with experience. Yes? Now, this capacity to adapt with experience is not constant through life. There are periods in life in which our brain is more effective to adapt with experience. Yes? So I'll give you examples. There are some very intuitive examples about that. For instance, language is one of them. Yes? So for most of you, I don't have to tell you that English is not my first language. Yes? You would pick, even if I come from Canada, you may think that I have somewhat a Canadian accent, but no. Yes? So even though I can communicate in English, yes, I didn't learn it early enough so that I speak it without an accent. All right? Okay, so that's one example of a brain function that is regulated early on in life. Yeah, we refer to these periods as critical periods. So for instance, here is you see that the capacity of the brain to distinguish foreign sounds decreases exponentially as a result of exposure of the brain to its maternal language. Now, one of the things that we learn over the last maybe 15, 20 years is that not only the capacity of the brain to regulate to functions such as language, yes, it's regulated through these critical periods, but also the capacity of the brain to regulate behavior, yes, and social responses. So this is work of a colleague of mine at McGill, Michael Meany, who spent his career studying maternal behavior in rats. So what Michael found is that variation in maternal behavior early on in life determines, yes, how the offspring develop. So the variation in maternal behavior that he measured is frequency of licking and grooming in rats. So he observed that rats naturally vary in the frequency that they lick and groom their pups. And he defined, one high licker and groomer group and low licker and groomers. When he looked at how the offspring from the high developed, he saw that the offspring are much more exploratory in their behaviors. So what he did is he took the pups from a high licker and groomer mom and cross-fostered them to a low licker and groomer, yes, within the first days of life, and he saw that it was really the maternal behavior that determined how they develop, but only if it happens early on in life. If he cross-fostered later, that was too late. Yes? So really, the early environment determines behavioral development in rats. So why? What happens? So there are changes that occur in the rats as a function of this maternal behavior at the molecular level. So for instance, one of the changes he observed is that the levels, the quantities of one receptor, known as the glucocorticoid receptor, changes according to maternal behavior. And what mechanisms can explain that? So let's go back to biology. Yeah? So this is biology 101, so that we're all on the same page here. Yes? So you remember, yes, that we have cells in our body. Our cells have, in their nucleus, they have DNA. Their DNA codes, have a code. Then of the code of the DNA, we produce RNA that goes from the nucleus into the cytoplasm, yes, and then is translated into a protein. So far, so good? Good. Now, our genetic material is organized within the cells, yes? So we have chromosomes, yes, that contain the DNA that is coiled around proteins known as histones, yes? That organization makes DNA more or less available to proteins in the cell that read the code of the DNA. Yes? All right. So now that we're all experts in biology, yes, let's just think that important ways that our organism regulates its genome, yes, and responds to environmental needs is by processes that we collectively refer to as epigenetic mechanisms. There are three main types of epigenetic mechanisms. There are those that are related to the chromatin or to the histone. Remember the proteins with which the DNA is coiled around. Those related to RNA, non-coding RNA or RNA interference, and then those related to DNA methylation. So I will talk, just for the sake of the today, about primarily this mechanism here of DNA methylation. Now let me just do a disclaimer here that it's really a simplification of the processes, yes? So you should take what I'm going to tell you tonight, as just with a grain of salt. It's just one example and it's overly simplified. So, DNAs contain genes, yes? Now, how do genes get activated? So typically, yes, there are transcription factors, which are proteins that identify certain sequences in the DNA. And then there are other proteins, such as RNA polymerase, that read the signal from the transcription factors, come here, and then would activate the sequence of the gene to produce an mRNA, which would then be translated into a protein. All right? Now, what happens with methylation, which is one of these processes that I told you about epigenetics, is that basically there are chemical changes that occur in the DNA sequence, yes, that interfere with the way the gene is read, that interfere with the way the gene is activated. And these methylation changes, these chemical changes that occur in the DNA sequence, in part, not exclusively, but in part, occur as a response to environmental factors. So when that happens, this methylation prevents the transcription factors from binding, or proteins to come here and bind and activate the sequence of the DNA. Yes? Now, remember, this is an oversimplification, but it's somewhat a way how these things happen. So we have learned, and this is work from Joe Acker's lab, that as our brain develops, the levels of methylation in neurons in particular, yes, change dramatically. As the neurons become more active and there is synaptogenesis, as we actually become more capable of functioning, yes, the brain changes significantly in its level of methylation at the neuronal level. So the hypothesis that we have had is that in part, yes, remember what I told you, is that we think that these differences in developmental changes that occur as a function of exposure to adversity are at the molecular level explained by changes in methylation. So how do we study that? So we study because we have access to brain tissue. Without access to brain tissue, there's nothing that we can do at this level because we need to study specifically, directly the brain. And so we have a brain bank, which is a collection of brains that we have in Montreal, whereby we receive brains from individuals who died from suicide or from other causes, and then we engage in a series of interviews with family members, with as well get information from youth protection services, from medical charts, etc., to understand the lives of the people on whom we have the brains. So for the studies that we did, or we continue to do, is typically we investigate individuals who died by suicide, were under foster care, under protection from youth protection, and had histories of severe childhood abuse. So I'm going to give you one example of how is that we think the environment or the exposure to adversity can change things in the brain and change behavior. And this has to do with an axis of that we refer to as the HP axis. So what is the HP axis? I'll give you one example. I was coming here tonight, and I took the wrong train. Yes? I got off the train. I realized I was late already. I realized I got the wrong train, and I look on the panel, and I said, where am I? And I thought, I'm going to be late for the talk. So my HP axis got really activated. It released cortisol from here, and I was in panic, yes, in a state of stress. So this is what the HP axis does. Yes, it helps us react to stressful situations and prepares for that. Now, after 5 minutes of panic, yes, I sort of say, okay, well, breathe in slowly, and then my HP axis got regulated. Why is that? Come down, cortisol goes to the hippocampus and binds into the glucocorticoid receptor and then inhibits, there's a negative feedback, we call it, inhibits the activity of the HP axis. All right? So the first study we did precisely looking at the genes that code for these receptors here that identify the cortisol being released and and then have a negative feedback on the HP axis. Why did we do that? Because we had evidence from animals that the glucocortical receptors regulated according to early life experience. So what we found was precisely that there were differences in methylation in this gene as a function of histories of adversity early on in life, of abuse during childhood. So in such a way that histories of adversity, of childhood abuse, basically increase significantly the level of methylation in the promoter, in the part of this gene that regulates its activity, which is precisely where transcription factors bind and activate the activity of the gene. Yes? So by having more methylation here, transcription factors couldn't activate the gene, therefore there was less quantity of this gene produced. All right? So far so good? Okay, so let me conclude. How do we interpret this finding? So we interpreted that here you have the HP axis. On your right, you would have a situation of exposure to a life adversity. On your left, you have normal development. Yes? So as I told you, as happened to me at the train station this morning, I activated my HP axis. The cortisol came here. Yes? It identified here the receptors, and then there was a negative feedback. So what is that we found in individuals who died by suicide with histories of adversity? We found that there was more methylation in the genes coding here. More methylation means less expression, less quantity, therefore less quantity of the receptors. So when cortisol is released, so it's less effective in suppressing the activity of the HP axis. So as a result, what happens is that the HP axis is more overactive, more hyperactive. And why would that be? So we think that that happens as a way to adapt. Yes? Why is so? Because often, as I told you initially, perpetrators of abuse are primary caregivers. But they're not always abusers. Sometimes they abuse, sometimes they not. Often the abuse comes associated with intoxication, yes, with use of substance. So the message the brain gets is that the environment is unpredictable. Sometimes it's hostile, sometimes it's not. Sometimes the person who is there to provide care does so well, and sometimes it does not. Yes? So the brain adapts to an environment that is unpredictable, that is hostile. And how does it do so? By increasing the levels of alertness. Yes? By being more vigilant, paying more attention. So levels of alertness means what? Means, at the clinical level, means being anxious. Yes? What does anxious mean? Is that you cannot switch off your brain. Yes? That you're constantly thinking, constantly alert. So this is one example of a possible biological mechanism that we think might help us understand the relationship between being abused during childhood and suicide risk, as well as other negative mental health outcomes. All right? So to conclude, I told you that early life adversity, yes, like exposure to childhood abuse, associates with changes in how the genes get regulated. And I gave you the example of methylation. Yes? So abuse would lead to changes in methylation in certain genes. These changes in methylation in genes would lead to changes in how these genes get transcribed, yes, in terms of gene expression. These changes in how these genes get transcribed lead to behavioral and emotional traits, and I gave you the example of how this may be associated with anxiety. There are other examples, but you have to invite me again another time to give. these changes, yes, then lead to other changes that basically sort of give a background, yes, that it's where I think is happening with individuals who were exposed to a life adversity and leads for them to be more at risk of all the consequences that I showed you initially in a slide. But specifically for suicide, I think that really the key is that when you have this background and then these individuals become depressed, with depression they have suicidal ideation, and in that case they have increased risk of suicide. All right, so I hope that was clear. If not, I'm happy to take questions. Before I end, I just want to acknowledge that the work that I do is work, that I do in collaboration with a lot of different people. This is a picture of my lab and of the research group that I direct. There's a lot of people involved in every different aspect of from the brain bank to the lab, etc., and all of them are extremely important for the work that we do together. Thank you very much.
Truly impressive, Dr. Turecki, and I think your groundbreaking work is taking us all by, not just by surprise, but gives us a lot of food for thought. You've provided a very broad overview, but also great insights into the impact of traumatic events in life of an individual, especially the early childhood or early stage of their lives, and how they may change the brain, and how those changes may lead to or predispose the individual to mental health problems, even suicide. Before we open to questions, I just might kick off with one question while our audience is prepared. Is there a treatment or intervention that can be given to modify these changes in the brain that are occurring even at molecular level that you've described? Or are these changes permanent?
All right, so thank you. So I think that the goal of this type of work is precisely to identify possible ways to intervene. Yes, today we have effective treatments, but they are not very specific, and they don't work on everybody. So we certainly need to increase our repertoire of treatments available to help people who were exposed to a life adversity. And the primary goal of this type of work is to understand what are the molecular mechanisms so that we can then really modify them. So where are we today in our capacity to modify these changes? So I would say that we have made progress. It is possible, technically possible today, to actually modify changes, epigenetic changes, or maybe not all of them, but certainly methylation. To modify them, we have to understand very precisely where these changes are located, in what specific cells, in what specific part of the brain, and this is information that we still don't know. And we have to be able to deliver the treatment and modify these changes effectively to the brain. It is possible to do it in cell models, and it's possible to do it in animals. Today, with viral vectors, for instance, we are able in animals to actually modify some of these epigenetic changes. But we're far away from doing this in humans, and in part because we still don't understand everything, or there's a lot of things that we don't understand. So we still have a long way to go.
I hope this gave you a few minutes to prepare. I will start with taking some questions. I think we have time for 2 or 3 questions. We have 2 roving microphones. Would you stand up and put your hand up so that the audience can see you? Now, I have to say I will be biased. I will start with a woman, because all research shows that if the first question comes from a man, the second and the third and the fourth will also come from a man. Proven, yeah? We'll start here. Thank you. There you go.
My question is the epigenetic change evident to pass on to future generations, or does it just stop with that generation?
So this is a very interesting question. So there is a whole field in epigenetics of what we refer to as transgenerational epigenetics, which is the study of transmission between generations of epigenetic changes. Of particular importance here is intergenerational transmission of acquired epigenetic changes, yes? Because otherwise, we have a number of epigenetic changes that are transmitted that we know, but are not necessarily acquired. So I would say there is interesting evidence that that happens. There's promising evidence, but it's not yet evidence that everybody agrees on it. Yes? So it's a very heated debate in the area, so to speak, about whether or not these changes actually are transmitted. There is evidence that they are transmitted at the phenotypic level. For instance, if there are studies in Holocaust survivors, for instance, that show how certain behaviors are transmitted. And in animals, there's data that shows that they are transmitted molecularly as well. Yes? But as I said, the judge is still out there as to whether they're actually transmitted.
Thanks. The research that you showed with the rats, with the nurturing, increased licking and nurturing, increasing the resilience and the lower level producing structural changes, then if you shifted those I think you call them infants, I don't know.
The rats?
Little rats, baby rats. When you shifted them from low lickers to high lickers early enough, they could increase their, I think, glucosteroids. I just wondered if there are any hypotheses around the age or the length of time for infants, young children, that you can get them out and sort of have a similar positive impact.
So basically your question is, what is the duration of this critical period where actually these changes are important? So the easy answer is no, we don't really understand, yes? And not only that we don't really understand, likely there's a lot of variability between individuals, yes? As to, what's the critical age that these things get regulated. Now, I want to be very clear in the fact that, our brain is constantly communicating with the environment, yes? And it's able to adapt to environmental experiences, yes? What I'm trying to propose to you is that its capacity, it's reduced because there are these changes at the molecular level that are more likely to occur in the beginning, but does not mean that later on they cannot change. For the same, if we make the parallel, for instance, with language, I think, I'm able to communicate even though I didn't learn English early on in my life, yes? And I may not speak with a perfect accent, but I'm still able to talk, I hope. So, yes? Now, it is, I think, the same, yes? And then likely it would change from person to person, its capacity to adapt, to regulate, behavioral and emotional responses later on as to a certain period. So it's a long answer to tell you that basically I don't know.
Thank you very much for the talk. I was wanting to know if you've been able to look at the effects on children who've experienced a lot of physical pain, who haven't been abused, but who've had medical conditions that have meant they've been in pain, whether there are similar effects on the methylation or not.
So that's a great question. So we have not, but I think my hunch would be that it wouldn't be the same, yes? For one of the things that I mentioned before, what it's really important is the psychological experience, yes? And it's the psychological experience. So we've done work, for instance, showing that in the risk of suicidal behavior is modified if the person is abused by a caregiver as opposed to by a stranger. Yes? So when there is abuse by a stranger, there's also an increased risk, but it's much higher if it is the abuse is given by a caregiver. Yes? So it would suggest that it's really the psychological experience that it's important and how the person feels about it.
Thank you very much. I enjoyed your talk very much. My name's Sam Ginsberg and I'm a clinical child psychologist. We've found with children with ADHD and with oppositional defiance disorder function very well when we change their environment from perhaps a cyber world and inside to sort of an outside, more natural environment, whether it's a playground, climbing trees, water, whatever. So I'm wondering if there's research at the moment having a look at the range of young children over the developmental spectrum and the extent to which being, if you like, in cyberspace and inside as a group, as against children who seem to show significant cognitive improvement and emotional improvement when they spend considerably more outside with Mother Nature.
Yes. So I think that, I cannot answer specifically about what you asked me, but what I can tell you is that what I'm presenting here is it's one side of the coin, yes? It's the sign of the coin of when things go wrong, yes? Now, I don't want you to leave here thinking that everybody who is exposed to abuse and has a history of maltreatment has negative mental health outcomes or die by suicide. There's a lot of people who are resilient to it, yes? But I don't, I study those that actually die by suicide, yes? So the results that I have associate with the people that were not resilient and were not able to actually cope and adapt to it in a way that they became functional, yes? So I am sure that people go through these experiences and necessarily they go and cope, and there are lots of people. Yes, in fact, there's a minority of the people who have histories of real life adversity that die by suicide. Yes, but I in order to study these mechanisms, I have access to the brain tissue. I need access to the brain tissue. Yes, and I can only do that in individuals who died.
I think, thank you. I think we may bring the questions to a close here. It is a fascinating topic. It is complex work. It is a theme that can really last tonight for a much longer time than what we have available, and it gives a lot of food for thought. On this note, may I please all ask you to join me in thanking Dr. Gustavo Turecki on an inspirational talk. I have to say that our annual Barbara Dicker Foundation Oration is not just an opportunity to invite inspirational world leaders in their chosen field who are making amazing social impact through their work. It is also an opportunity for us to add another wonderful individual to the Swinburne family. And Dr. Gustavo Turecki, you're a new member of the Swinburne family, and we look forward to working with you closely over the years through our institutes, through our centers, through our leading researchers, but as well as PhD students and early career researchers who are the future workforce that will carry on this wonderful work. Thank you. Ladies and gentlemen, this concludes our evening, and I would like to once again thank you for joining us. I encourage you to keep an eye out for our Swinburne alumni newsletters and websites so you can stay up to date on the cutting-edge research and innovations coming out of this wonderful institution. And follow, please, the exciting outcomes that are produced daily by our students, our staff, our researchers, educators, and so on. I look forward to welcoming you to other events in the future and continuing to see our Swinburne community that addresses the challenge, community leading the way in world-class research and innovation that addresses the challenges of our time. Thank you once again, and I wish you a wonderful evening.